In this lecture, we will be discussing the processes of distribution and some metabolism. Distribution is the process in which an absorbed chemical moves away from the site of absorption to other areas of the body. So some of the questions we'll be looking at today are: how do chemicals move through the body? Does distribution vary with the route of exposure? Is a chemical distributed evenly to all organs or tissues? How fast is a chemical distributed? And why do chemicals stay in the body for a long time while others are eliminated quickly? The diagram on the slide depicts the various compartments in the body and how a chemical from the outside environment might get absorbed and distributed within these compartments. It also tells us a bit about the volumes of these compartments, and therefore it can inform us about the likely concentration of a compound if we know the initial dose. The first compartment we will mention is the plasma, which is the liquid part of blood, as this is the compartment where the toxicant will likely travel in to the rest of the body. In addition to this, there is the interstitial space. This is the space within organs and tissues that is not occupied by individual cells. Finally, there is the intercellular space. This is the combined internal volume within all the individual cells of the body. When we think about the distribution of a toxicant, we need to consider all of these compartments in order to discuss its toxicity and target organs. If we consider an individual chemical for a moment, following absorption, chemicals enter the bloodstream. They get diluted and will be distributed around the body. It can be metabolized and then get eliminated by excretion. This is illustrated in the flow chart in the slide. The extent and the duration of these processes will vary from chemical to chemical. The plasma concentration of a chemical often directly relates to its concentration at the site of action. The total body water that we saw in the previous slide may be divided into plasma, interstitial, and intracellular water, and these all contribute to the concentration of a toxicant. The volume of distribution describes the distribution of a toxicant between plasma and tissues. Another way to describe it is the volume in which the amount of chemical would need to be uniformly dissolved in order to produce the observed blood concentration. The formula we have for this is VD, which is volume of distribution measured in litres, is equal to the dose in milligrams divided by the plasma concentration in milligrams per litre. If we look at our pet toxicant worked example, paracetamol, for a given 1000 milligram dose and a peak plasma concentration of 20.8 plus or minus 6.9 milligrams per litre, measured 48 minutes after its ingestion, the volume of distribution is 48.1 litres. So what does this mean? This can all be related back to the total body water and what compartments it might find itself in. If the volume of distribution is equal to the plasma's volume, which is 4 litres for example, and the dose is 1000 microlitres, then the plasma concentration would be 250 milligrams per litre. If, however, the volume of distribution is greater than 42 litres, it means that the toxicant has distributed to the tissues and very little is in the plasma. As a reminder from the first lecture, bioavailability is the fraction of an administered dose of unchanged drug that reaches the systemic circulation. When a drug is given intravenously, for example, then the viability is 100% because all of it is immediately reaching the systemic circulation. If exposure is by other routes, oral for example, then bioavailability is decreased due to the incomplete absorption, first-pass metabolism, or other factors. The bioavailability of our worked example paracetamol is between 75 and 90% for oral delivery. If we look at some of the data on paracetamol oral dosing published in a study from a number of years ago, we can see that a 1000 milligram dose was given orally, and the graphs shown in the slide show the levels of paracetamol, or acetaminophen as it's sometimes known as APAP, in the plasma on the left, and the levels of its metabolites on the right over 6 hours in 5 healthy volunteers. As we can see, there is a peak plasma concentration of around 20 micrograms per mil just before the 1 hour mark on the graph, and this decreased then over time. At the same time, the concentration of the various metabolites shown in the graph on the right increased more slowly and peaked at 2 hours and then are decreased over time. From measuring the levels of the parent molecule and the metabolites, we can then estimate the volume of distribution and determine the extent of its distribution. So what factors can actually affect toxicant distribution? Firstly, the site of absorption can influence the degree of metabolism, storage and elimination of toxicants. Substances taken orally generally go into the liver, portal circulation, having been absorbed from the gastrointestinal tract, and undergo first-pass metabolism. But absorption via the skin or the lungs, they go directly into the circulation where there is generally very little or negligible metabolism. The amount of toxicant a tissue or organ receives depends on the volume of blood to that tissue. Well-perfused lean tissues include the heart, the lungs, the liver and the brain. Moderately perfused lean tissue include the skin and the muscle. And finally, fat and also negligibly perfused tissue include the bones, teeth, tendons and ligaments. Plasma protein binding leads to formation of a protein-toxin complex. As a result, distribution is limited, its half-life is prolonged, and excretion is also limited. Finally, storage of toxicants can occur in fat, in bone, in liver, and in kidneys, and the extent of this depends on the chemical nature of the toxicant. So we've mentioned there about plasma protein binding. What is it, and how does it affect distribution? Well, plasma protein binding facilitates the transport of poorly water-soluble compounds. It therefore reduces the concentration of the free compound in the plasma because it's actually bound to something. It also restricts the distribution as only unbound chemicals will cross membranes and distribute into tissues. The reason it restricts excretion is that plasma protein bindings, albumin included, are too large for renal excretion. Binding sites on plasma proteins can become saturated, and small changes in the dose therefore can cause a large jump in their plasma concentration. Different types of toxicant can even compete for the same binding site, and this can lead to displacement, causing an increase in the free plasma concentration of one or both of the chemicals. Rates of distribution to tissues and organs involves the passage across membranes as shown in the diagram on the top right. It is affected by the same physical, chemical factors as absorption, such as the size, shape of the molecule, its lipid solubility, hydrophobicity, its charge and polarity, its structural similarity to endogenous molecules. Distribution to tissues is mainly by passive diffusion, meaning along a concentration gradient, and involves the passage out of capillaries, the extent of which depends mainly on its size and its lipid philicity, as we discussed the first day with regards to absorption. This is possible as there is a discontinuous barrier in most organs, as shown in the diagram on the bottom right. The rate of distribution depends on blood flow through the tissue. High flow rate means a rapid delivery to organ or a tissue, and compounds with some water solubility will be distributed throughout the body. In some cases, lipophilic compounds preferentially distribute to fat, for example, the dioxins DDT and TCDD. Other compounds accumulate in specific tissues, for example, like lead in bone because it mimics calcium, paraquat in the lungs because there is uptake by a specific transporter, carbon monoxide in red blood cells because it's able to bind more preferentially to hemoglobin than oxygen, cadmium in the liver and kidneys because it binds to a specific protein called methylmethionine. Toxicants are often concentrated in a specific tissue, which may or may not be their site of toxic action. Toxicants can compete with and displace endogenous molecules from plasma proteins. Lipid-soluble toxicants can accumulate mostly in subcutaneous adipose tissue, and toxicity can therefore appear less severe in obese people. Also, the exchange between the extracellular fluid and hydroxyapatite on the bone surface can mean that several chemicals can compete and substitute for calcium and hydroxide ions in the bone matrix, like lead, as we've mentioned already. As a result, the biological half-life of stored toxicants can be very, very long, and this can change though with periods of starvation, which can lead to the release of stored toxicants from fat, for example. The brain has an extensive blood flow, which is roughly about 16% of the total cardiac output. There is understandably a restricted entry of xenobiotics or foreign chemicals. If we compare the top and bottom diagrams on the slide, we can see that there is reduced permeability of brain capillaries compared to those of normal tissues. In the brain, there are what are called tight junctions between the endothelial cells, where there are few pores and a continuous basement membrane. This means that entry is restricted to non-polar lipophilic chemicals by passive diffusion. So the next question that concerns us is what happens to toxicants once they reach a tissue or organ? Well, most chemicals absorbed in the body are lipophilic, as we've already seen, and are not well excreted in the urine and or bile due to reabsorption. But biotransformation is an enzyme-mediated transformation of compounds, both endogenous and exogenous, and ends up converting lipophilic compounds to more hydrophilic or polar compounds, and results in more polar metabolites, increases in molecular weight sometimes and size, and definitely facilitates their excretion. The consequences of these processes is that it reduces the half-life of the parent compound, reduces the duration of exposure and therefore toxicity, it avoids accumulation in tissues and organs, it changes the biological activity of these xenobiotics. The process itself of biotransformation is carried out by a limited number of enzymes that have broad substrate specificities. There are two phases of reactions in biotransformation. Phase 1 reactions involve the introduction or the unmasking of a functional group, as shown in the diagram on the bottom of the slide. These types of reactions include oxidation, reduction, hydrolysis, and hydration. Phase 2 reactions, on the other hand, involve conjugation with larger, more polar or water-soluble complexes to increase the solubility and facilitate the excretion via the urine or bile. These include reactions like glucuronidation, sulfation, acetylation, glutathione conjugation, methylation, and amino acid conjugation, and will be discussed in the next lecture. Metabolites may be more toxic than their parent compounds, especially metabolites from phase 1 reactions. Metabolites can be more reactive than their parent compounds, and this is a process known as metabolic activation or bioactivation. It is important to understand that biotransformation is not linear and not a sequential process, meaning that it does not have to occur phase 1 first, phase 2 second. It involves multiple reactions and can involve multiple different enzymes. Phase 1 reactions are mainly catalyzed by what are known as monooxygenase enzymes located in the endoplasmic reticulum and are called microsomal enzymes. These include cytochrome P450 family and FAD-containing monooxygenases. An example of a phase 1 enzyme is alcohol dehydrogenase, or ADH for short. It is a cytosolic enzyme present in the kidney, lung, and gastrointestinal tract, but its highest activity is present in the liver. Shown below are reactions catalyzed by alcohol dehydrogenase, main one being the conversion of ethanol to acetaldehyde and methanol to formaldehyde. A second enzyme that we'll look at here is the enzyme aldehyde dehydrogenase, or ALDH. This is present in the cytosol and the mitochondria and oxidizes aldehydes to carboxylic acids, with NAD+ used as a cofactor. Shown below are the reactions catalyzed by aldehyde dehydrogenase converting acetaldehyde to acetic acid and formaldehyde to formic acid. Monoamine oxidases is another group of phase 1 enzymes, and it's involved in oxidative deamination of primary, secondary, and tertiary amines, including serotonin and a number of differentiated biotics. Oxidative deamination of primary amines produces something called ammonia and an aldehyde. Secondary produces a primary amine and an aldehyde. Monoamine oxidase is located throughout the brain and in the outer membrane of the mitochondria of the liver, kidneys, and blood platelets. Phase 1 reduction reactions are also carried out by microsomal and cytosolic enzymes, such as the cytochrome P450 family, the aldoketo reductases, and short-chain dehydrogenases or reductases or SDRs. Metals and xenobiotics containing an aldehyde ketone, a disulfide group, a quinone, an alkene, or a nitro group end up being reduced. Aldoketo reactions involve reductions to primary alcohols and of ketones to secondary alcohols that are catalyzed by these SDR enzymes. Quinones can be reduced to hydroquinones by two cytosolic flavoproteins known as NQO1 and NQO2, and both have different substrate specificities. So, one of the major contributors to biotransformations are the cytochrome P450 family of enzymes, so called from the different spectrum of the CO complex of reduced cytochrome P450, where P stands for pigment, 450 stands for 450 nanometers, the wavelength at which it's absorbing light. This family are located in the smooth endoplasmic reticulum and are also known as microsomal enzymes. They contain an iron protoporphyrin ring, as is shown in the diagram on the right, and they use this as a prosthetic group. They are closely associated with NADPH cytochrome reductase and you transfer 2 electrons to the P450 molecule. They are a family and a collection of isoenzymes, meaning that they are one of several different forms of an enzyme. In particular, genetics and the environment can actually influence their activity. Shown in this slide is an overview of some of this family of enzymes and an explanation of their naming system. These are broken down to family and subfamily levels and follow a similar pattern in naming by using a number, a letter, and then a number finally. Examples of individual enzymes and their substrates are given at the bottom of the slide, for example, CYP1A2 and paracetamol being its substrate. If we apply the information we learned today to our pet toxicant work example, paracetamol, we can see that under the bioavailability, plasma half-life, volume of distribution, and metabolic enzymes that this information has now been added.